Good morning, good afternoon, and good evening, everyone. It's a great honor to welcome you all today to the session of Proton Lecture Series. We are privileged to have with us our the estimate expert in ophthalmology, Dr. Ranju Kharel, who who will be sharing her insight in SHAPU in her talk about SHAPU. She is an associate professor and uveitis specialist at the BP Koirala Line Center for Ophthalmic Studies, Institute of Medicine of Nepal. She is also associate professor of ophthalmology at the Institute of Medicine, at the Tribhuvan University. Dr. Kharel is dedicated to SHAPU in Nepal. She is one of the lead experts in this topic. And today we have the great honor to have her here giving us this lecture. Please join me in welcoming Dr. Ranju Kharel to this talk.
1 second, Dr. Ranju, and hi. Thanks everyone, and thanks Carlos for introducing Dr. Ranju. And dear friends, I think it's a great honor. And for those who don't know Dr. Ranju's work, along with Dr. Madan Upadhyay, who's on the call, and they have been phenomenally working in the field of uveitis, and particularly for a specific disease called as SAPU. And Dr. Ranju, in next an hour or so, she will explain to you what is SAPU and why it is so important and the work. But I think she really deserve a big applaud for the efforts she has put in this particular field. I think this is phenomenal. This is unparalleled, and with very limited resources at her disposal, the efforts she has put in to make this field really something at a global level is something phenomenal. And I myself witnessed the efforts she has been putting under the mentorship of Dr. Upadhyay. So I think incredible amount of work, and I'm honored to have her here, and we Proton Group is honored to have her here. And she is one of the main collaborator and advisor for Proton Group. So thanks, Dr. Anju, for sparing Friday evening with us and then giving our talks to us. Thank you.
Thank you so much. I sincerely appreciate the opportunity to be a part of this Proton Lecture series and share insights in One Health in Ophthalmology, the case of SHAPU. It's indeed an honor to engage with such an esteemed group and contribute to this important lecture series. Thank you, Dr. Rupesh, Dr. William, Dr. Carlos, and the whole team. Your dedication in fostering knowledge exchange and interdisciplinary collaboration is truly commendable. So I think without further ado, I begin my presentation. May I, Dr. Rupesh?
Yes, please.
So I think it's full screen.
Yeah.
Yes, doctor.
Let me begin to this topic in Proton Lecture series. And first of all, I would like to disclaim that all the pictures that are being used in this presentation, they belong to our own work. So nothing taken from Wave or anywhere else. And for many of the people, this SHAPU could be a first-time hearing word, but this each letter in this word, it has some special meaning. Like the S means seasonal, H means hyper, A means acute, P means pan, and U means uveitis. So it's a particular type of uveitis that is hyperacute in nature and seasonal in pattern. And as said earlier, yes, it's indeed a mysterious uveitic conditions, especially in Nepal, because it not only because it takes the sight of the person, but also makes them cosmetically blind, cosmetically disfigured, like this child who has grown up not only losing sight in one eye, but also being cosmetically disfigured because of the shrunken thigh globe. Yes, we are here for this peculiar disease because it bears a lot of peculiarities, and few of the peculiarities that I want to share at the beginning are it usually occurs as an outbreak in Nepal in the odd years. The first outbreak was noted in 1975, the next outbreak occurred in 77, 79. So in a sequential odd year pattern, the last outbreak was in 2023, and now the next expected outbreak is in 2025. Yes, We're already in 25, but the outbreak won't occur throughout the year because it has a seasonal pattern, and that pattern is after the monsoon season. That's in the autumn season of Nepal. So it will occur. It is expected to occur as an outbreak from August onward, and it can affect any age, any gender, but predominantly it affects the children. But surprisingly, unlike other uveitis, it has all the time unilateral presentation. Both the pictures, they show that these are the child and they have unilateral presentation. Uveitis by default, they are supposed to have chronicity or recurrence, but this is a one-time disease, one eye disease and one-time disease. So disease uveitis without no recurrence. Either the site is saved or the site is gone and the cosmos is also gone. So it's a one-time disease, but however, it is very sudden in onset, and many of the time it gets confused with conjunctivitis, but it does not have discharge, though the redness is present. But it do have profound loss of vision, and unlike other causes of leukocoria, this is red eye with white pupil. So not white eye with white pupil like in retinoblastoma or congenital cataract. It is red eye with white pupil, and the eye is very soft. That is called as malignant hypertension. And this disease especially has special affiliations with the white moth. This disease is mainly reported from Nepal, but we recently have two reports from Bhutan also. Now, for more difficult part for us is like this disease, we exactly don't know what is the pathogenesis for this disease. So we are treating the patient with a blanket therapy covering the infective and the immunological arm. So when the outbreak occur, the patients they come in clusters. So these are the clusters in different outbreak, and the clusters were high when we were having the second outbreak of COVID in 2021. So in 2023 also we had received patients in clusters. So managing each case is so difficult, and the cluster management becomes more chaos for all of us. Now, in our today's topic, where we are talking about One Health in SHAPU, I'll be describing the presentation in two part, the past and the present situation where we took the journey towards One Health. So in the past, I'll be talking about from the journey from 1975 to 2016, and in the present from 2017 onwards till date. So let's move on to past for the time being. So that past journey was the longest journey of 41 years. And in that time, we found that when we started looking on what actually happened at the time of inception, we found that the at first in 1977 75, few school students in Pokhara is a tourist place in Nepal. So few students they had redness, then white eye, then blindness. So at that time, there were very few ophthalmologists in Nepal, only 5 or 6 ophthalmologists, and Dr. Malla was one of them. And he had recently come back from UK, and he thought that it's a kind of it was occurring in a particular season, and it's a kind of seasonal endophthalmitis, and he found few moths around it. So he thought that it could be associated with tussock moth. And later on, Dr. Madan Upadhyay, who was 35 years at that time, he observed the same pattern in 75, 77, 79, 81, and 83. So with this consecutive observation of similar pattern of disease in similar location of Nepal and similar features, he named the disease as seasonal hyperacute panuveitis, that is SHAPU. That's why the name termed as SHAPU, and luckily, this SAPU was one of the topics that was presented in the first IUSC meeting that happened in 1984 in Tampere, Finland. So for his contribution in SHAPU, this coining and works on SHAPU, we call Dr. Upadhyay as the father of SHAPU. Yes, some people think that it could be endophthalmitis, but though it is similar, it is not endophthalmitis because the spread of inflammation in SHAPU is from anterior to posterior. First is the anterior chamber that is involved, not the vitritis, as in case of endophthalmitis. There's lack of pain or very minimal pain, lack of discharge. The presentation is very rapid, and to label it as endophthalmitis, like endogenous endophthalmitis, there are no evidence of being immunocompromised, no evidence of systemic disease. And to label it as exogenous endophthalmitis, there is no evidence of trauma. So this looks like endophthalmitis, but this is a separate entity and not endophthalmitis. So what happened during that past era was studies were conducted under the mentorship of Dr. Upadhyay, and luckily Professor Nusenblatt was also here in Nepal during that period, and even experiments with the animal models were also performed. So that was the first largest case series studies, which was published in 1984. Then a first report on detection of varicella zoster was done by Dr. Biswas from Sankhanetrale, where they had detected varicella zoster in immunofluorescence, and that was a patient from Nepal who had visited Sankhanetrale. The first report with association of SHAPU with bacteria like Streptoneumoniae, Staphylococcus, and Acinetobacter was made by Dr. Anumanandan in few subsequent outbreaks. And the first PCR-proven association of the SAPU with anellovirus, that is a toctenovirus, was done in 2011 outbreak where the samples were taken to Rotterdam. So there were variation in presentation, and this was the first largest series study where the samples were taken with the support of Dr. Nushin Blad to Procter Foundation, and they were analyzed to rule out other causes of leukocoria and uveitis. So when we first started our journey and we looked back, we found that there were only 10 papers over the past 41 years on SHAPU. So this SHAPU was more of a localized disease, and this was one of the landmark paper. And the information to the global ophthalmologists were limited. That's why we started our present journey in SHAPU from 2017. So in 2016 December end, I returned back from Sankhanetrale with after the uveitis fellowship, and then under the mentorship of Dr. Upadhyay and other ophthalmologists, we started our journey in SHAPU. But the journey was not that easy because there were a lot of challenges in front, where to move ahead. The first thing that we thought was let's make this disease a globalized. So the first step that we took in 2017 was entering SHAPU into a global platform like iWiki in AAO. We also reviewed what were the existing literature. So we published a 40 years review paper of SHAPU on 2018. And the main issue was with us was like the patient used to come with the history of contact with moth, but we didn't have a evidence best to prove it. So under the support of WHO, we did a nationwide study to understand the various biological and behavioral risk factors associated with SHAPU, and this was a case control study 1:3 ratio, where the cases that were seen in 2017 outbreak, the controls were tracked back into their geographical locations, age and gender matched, and we looked into all the risk factors, and we found that the contact with the moths, they were the primary risk factors in univariate analysis, and this was proven by the multivariate analysis also. So the odds of getting SAPU after having a direct or indirect contact with moths were 6.8 times higher. So these are the moths that we are talking about. So there is difference between a moth and a butterfly. Butterflies are the one which are beautiful in pattern, which come in daylight vision. The moths are the one which come in night light vision. So these are nocturnal. So these moth with white moth with black striations were the suspected culprit for the disease. This we got finally published in ophthalmic epidemiology. So these are the moths which are attracted towards the white light in the night time. So the photo on the left hand side, we can see that there was a pillar in the eye hospital where under the influence of white light the moths were resting on the wall. So this is how a female moth is laying egg, and then we came to know that these browns are the setae and the white are the eggs that are laid. So this setae, they form a base or platform for the eggs to rest. So these setae are laid by the female white moth to protect their eggs. But these setaes are their defensive mechanism, and they are toxic when they come into contact with we humans. These setaes are very friable, very easily airborne, and they can travel anywhere, so we don't need to directly touch the moth. The cities can be blown anywhere, they can come to our hair, they can rest in our tables or towels anywhere, so we can have an indirect contact with them anytime. So we went close to those cities and egg, and we could also capture the picture of each of these eggs and undergo test for them. So let me revise. These white are the eggs, the browns are the cities, and the culprit that we believe for SHAPU are the cities that are there for the protection of egg, but they are toxic to the humans. And during this course, we found that the youngest affected SHAPU case is a 30 days old child, where the mother was breastfeeding her child, and the moth that was in the light fell over the blanket. We could also prove the presence of moth hair in the corneal tissues and published it in ISAO. Surprisingly, the main surprise comes here when we see the presence of moth hair or cities in the ocular surface, in the corneas, or in the aqueous. Though the cities are present on both the corneal surfaces, the only one eye is affected. So that it's a unilateral disease. That's very surprising. The city could be present in both the eyes, but the disease is present only in one eye. So this is how we ophthalmologists we joined a new arm in this disease, and the human eye got connected to the moth or insect animal that or insect in form of moth. So this was our first step in One Health, where we ophthalmologists joined with entomologists to undergo further studies on SHAPU. So this is our team of entomologists where we work together. Now, this was just the first step, because with the moth came a lot of questions. We thought that there would be three schools of thought for this disease condition. The first one, are these cities are releasing some toxins or venoms that are causing hyperacute response? So is SAPO a toxicological buzz? That was our first question. Next, we thought maybe the moths are just acting as a vector for transmission of some microbes to the intact surface, or they are just acting as a secondary offender, some commensal organism getting pathogenic from the ocular surface, and is SAPU or infectious buzz. Our third hypothesis was, is SAPU just a cross antigen reactions, like where the moth and the feces are just acting as a uvitogenic agent? So is SAPU or inflammatory buzz? So we had to head ahead with these three schools of thought, and there were evidence to prove each of them. That was the more difficult part for us. I had shown you this picture earlier, but did you notice there is presence of rash? There is red eye with white pupil, or white eye with red pupil, whatever. Red eye with white pupil, or white pupil with red eye, whatever you say, and some rashes around the face area. So is it like something like when we come contact with some caterpillars or some insect toxins, we can develop rashes? So is it something like this? And this picture is from one of our resident who was wearing apron, and accidentally there was a presence of white moth in that apron, and when he removed, we found the presence of rashes in his neck surface. Luckily, he didn't get SHAPU. So there are evidence which says that these cities, they contain chemicals like acetylcholine, formic acid, histamine, trypsin, chymotrypsin, some proteolytic, hemolytic, and anticomplementive agent, which could cause harm to to any of the surface, and it could be the ocular surface also. So that was the toxicological buzz, but there were evidence to support the inflammatory buzz also, because when we try to undergo the studies on the analysis of interleukins like interleukin 6 and 10 from the vitreous samples, we found that there was cytokine storm within the eye, and interleukin 6 was highly raised. So this was evidence that there is inflammation, high grade of inflammation with ciliary shutdown is going on within the SHAPU affected eye. And another support to label it as inflammatory buzz is that we tried the treatment with two steroids and one antibiotics. So a short-acting steroid like dexamethasone, which can suddenly control the intraocular inflammation, and a long-acting steroid like triamcinolone acetonoid, which can maintain the intraocular pressure with the add-on moxifloxacin. So we tried the CDM sort in 2023 outbreak, and we had very good outcomes. So we have equal evidence to label it as inflammatory buzz. Finally, there are supports to label it as infectious buzz also, because these blue are the ones bacteria that have been detected from the ocular samples of SHAPU patient. This orange are the virus that have been also been detected from the ocular samples of the SHAPU patients, and even fungus have been detected. So some of these detections are from culture, some of them are from PCR, and some of them are from metagenomic. But not a single organism have been detected every time. So despite our limited resources, we did try with metagenomic analysis in 2019, and 2 out of 30 samples. Now, 2 out of 30 vitreous samples, we found that there was presence of streptopneumoniae in 2 samples and Staphylococcus epidermidis in 2 samples. But at this time, we couldn't label this as the sole cause of SHAPU because we lacked the report of the normal conjunctival flora. That was the limitation of the study. Then subsequently in 2021, we redid this metagenomic new generation sequencing, but this time the result was different because it was not just the streptopneumonia or staph epidermidis. It is this time we found that the main hits were high for the Acinetobacter barisini, and we rarely have heard of any UVIT condition which is so hyperacute in the situation caused by Acinetobacter barisini. And more surprise was that the eye which in where the Acinetobacter barisini was detected, that eye also the PCR was also proven for HSV, human herpes virus 7, that is very uncommon, not like herpes simplex or varicella zoster, that was also detected. So in the same eye, Acinetobacter and HSV7 was detected. So this time also we couldn't just label this as infectious etiology. Next, in recent 2023 outbreak, we again ran the metagenomic analysis, and this time we found that of 5 samples out of 35 samples, they are positive for streptopneumoniae and also for Parvovirus organism. But surprisingly, the not only the vitreous sample, the affected conjunctival swab and the unaffected conjunctival swab, they were also positive for the same streptopneumoniae variant. So it is hard for us to prove that whether these were the common cells that got activated because of some trigger or they are truly the sole cause of disease. That's why we are not in a situation where we can just label it as a pro-infective condition or a pro-inflammatory condition. But we believe it could be a pro-infective with a secondary inflammation or more likely a pro-inflammatory condition with a secondary infection. We yet don't know which came first. So we had recently published our paper on advanced diagnostic technology in SHAPU, where we have labeled mentioned both of this result. Now, one of the biggest lacuna in this SHAPU journey was the absence of classification system in SHAPU. So after 46 years of its first identification, we could develop the SHAPU classification system, and we could develop as mild SHAPU where there are very minimal inflammation. It looks like a normal eye. The anterior chamber has very few cells, and the vitreous is also relatively clear. But within 48 hours, there could be presence of hypopyon in the anterior chamber and anterior vitritis. And even if it is ignored in that time, thinking at conjunctivitis or any other form of uveitis, there could be presence of severe reaction from anterior to posterior part of the eye with malignant hypotension and retinal detachment also. So the progression from mild to severe can be in 2 to 7 days. And we then thereby came with this classification system of SHAPU as mild, moderate, severe, and we also established the SHAPU inclusion criteria. And this classification system helps us to at least form a treatment protocol where the mild cases can be treated with a topical medication. In the moderate case, we need to do the tapping, like the pus is coming out of the vitreous in tap, and then we give the intravitreal injection, and in the severe case, definitely a complex vitreoretinal surgery is needed. So this got published in Ocular Immunology in 2021. Now our life are at risk because we go to forest at night during the outbreak time, where we tie up this white net and white cloths and then lit the white light, and then we wait for the moths to come there, and these are how the moths come in the net. We collect them in the jar, then we bring to the lab and we pin them up and we do the studies on them. So just being an ophthalmologist is not enough for cases of SHAPU. So we needed what is the name of this moth, what is the taxonomical identification, and then we found that these are the moth which belong to family Notodenti, and these moth are Gazelina moth. So then we further underwent to study its species, and we found that there are four variants of Gazelina: Chrysolopha, Apsara, Transversa, and Intermixa. And the ones that are predominantly found in Nepal are the Gazelina Chrysolopha. This Gazelina Chrysolopha, they are found in Pakistan, England, and some other part also. But why the SHAPU is not there in that terrain, maybe I'll be able to answer you in the coming slides. So these Gazelina chrysolopa, the one with the yellow heads are the male versions. The one with the yellow tails are the female version, this yellow tail. So these are the places where the cities are present. The cities are connected to the abdomen. They contain some fluid within their city, and the cities are there for the protection of the egg. So these are how the moth they appear in that particular season in our environment. So huge number of moths, and when we look closely, we see that the males and the females are together. This is another example where the moths are like densely present in the glass, in the walls during that particular season, when the SHAPU outbreak occurs. So our next step was to identify where are the distribution areas of this Gazelina moth in Nepal, especially the Gazelina chrysolopha. And then let me say that this rectangular is the map of Nepal. We are surrounded by India on three sides and China on the top side. And when we studied the distribution of Gazelina chrysolopha in Nepal, we found that it is in the mid Nepal, above 200 meters from the sea level, not towards the tropical regions, but in the subalpine and subtropical regions. And then not it didn't end just with the distribution. We started undergoing its life cycle studies. So we started rating the moth from the eggs to the five larval instar. So this is first, second, third, fourth. The fifth larval instar has the highest life cycle, and then it comes to pupa, the adult form, and then by August, September, the adult form they emerge in the environment. So these are the larval form, and these are like we were rearing inside our lab, and we found that they had to depend upon some food source to grow. And what were those food source? Those were the leaves of these plants. So here came the next arm of One Health in ophthalmology. So these trees were very important for the survival of this white moths, and these trees were nothing more than the special plant that are more specifically found in Nepal, and it's called as Alnus nepalensis. So we could add the next arm in One Health in ophthalmology. So along with the eye moth, the atlas came to play into role for the acquisition of SHAPU, and we added the forestry experts in our team. And then we started looking for this moth, and then we found that the initial caterpillar, this larval phase, they are the processionary moth. So they walk in processions, so like in a line form, and they eat the leaf of this Alnus nepalensis for their completion of life cycle, for their growth. So it is very important for these plants to be present in that particular area for the completion of the life cycle of this moth. And so our next step was to look into the distribution of these plants in Nepal, and we found that these plants were also distributed in a similar ecological belt like that of those moths. And we also looked into the microhabitat of both this host plant and the host moth. What were the suitable humidity, the suitable temperature, and how close the human habitat towards the endless forest is like prone for SHAPU development. And we found that they were closely connected. There has to be presence of that particular temperature. So that particular bioclimatic variation was important, and the placement near that Alnas habitat, Alnas forest was also important for the disease to occur. So, and more importantly, that rainfall, the precipitation in in that climate, in that season month is also important. So this all has to come together for the SHAPU to occur, because these are the host plant and the host month. So all these things might not come together in other part of the world. So that is particularly in Nepal, in particular terrain of Nepal. So Alnus nepalensis, as the Nepalensis would say that these trees are very specific to Nepal. So when we study the distribution of SHAPU in Nepal, we found that again these are the mid mid mountains and mid hill regions in Nepal, not towards the border side of India where it is a hot climate, not towards the mountain side where it is a very colder form. So that particular terrain, that particular ecological dynamics was very important, and we found that this ecological dynamics of Nepal was quite similar with the Bhutan also. The same Hindu Kush mountains, the same ecological, the same climate ecological similarity that could have made the presence of SHAPU in Bhutan. Moreover, those ophthalmologists who detected SHAPU in Bhutan, they were the ones who were trained in Nepal, who did the residency in Nepal, so they had known about the characteristic of this disease in our country. So when we finally completed the One Health in SHAPU by adding the ecological arm. So this time we could make a final team of ophthalmologists with entomologists, with forestry expert and the ecologist, because everything has to come in place for the disease to occur. So this was our first One Health group, which was made under the guidance of WHO and Nepal government. And then we started conducting different workshops, taking all the stakeholders so that we can understand the disease, because just being a mere ophthalmologist is not being, it will not be enough to solve the mysteries that are related with the SHAPU. So one more thing is that we as a human, we have equal, the right that we have to live in this planet. Maybe all the animals and the plants, they also have the same right. So this planet belongs to all and it is the one health system for all. But when there is a combination of that particular host plant, the particular susceptible host human, and the particular host moth, then the disease can happen in form of SHAPU, the blinding eye disease. So this was an ecological model that was developed way back before we could finalize all our arms. This model was developed by Dr. Upadhyay in 2-3 years back, where he had already described that there has to be this plant, the moth, and the human, and the human host should have a particular genotype or phenotype and their immune status to be affected with the SHAPU. Both the child from a single family may be exposed with the same amount of moths, the same amount of ecology, but we find that only one of the siblings is getting the disease, the other one is being spared. So that has to be done with certain genotypic um, characteristic. And after having SHAPU, there is a huge gap in understanding what is the mental status, what is their educational status, and what is the quality of life in such individuals. So let me once again revise this. These yellows are the distribution of Gazelina chrysolova in Nepal. These green ones are the distribution of Alnus nepalensis in Nepal. So these red ones are the distribution of SAPU cases in Nepal. So when we stack over the things, all this has to come in a common place for the disease occurrence. That's why SAPU is a disease which is not found throughout the country, but in specific regions of Nepal, and it's not found in the areas which are close to India or close to China, but it is very in a localized regions. Now, with all these things in behind, maybe very soon in future we'll be proposing the new name of this condition as uveitis nepalensis, because this is a disease very specific to Nepal. And this is a classical examination. This is a classical picture of red eye with white pupil. And let me repeat that this white pupil is not because of the cataract. It is because of the dense exudation that is present behind the lens, causing the whitish pupillary reflex. Now we could achieve up to that label, but we have a lot of challenges in front because we yet don't know its exact etiopathogenesis. So in lack of that, we are not able to develop the treatment protocol. We are yet to confirm which are the genetically predisposed people for developing SHAPU. and our awareness and preparedness across the countries, it's ongoing, but we have a very difficult terrains. We have high amounts of Himalayas and mountains, so reaching everywhere is very difficult. And those patients who accidentally have SHAPU, for them to reach to the eye hospitals on time is also very challenging. Maybe they have to cross some 2-3 hills. They might need 2-3 days to reach the primary eye health center. So that's a challenge, and this duration of 2 days might lead the case to the severe stage where the visual prognosis is very limited. And we are yet to understand the quality of life and vision in them. And now recently we found that these SAPU cases are involving in the newer regions of Nepal, maybe because of this ongoing global climate changes. It's SAPU is expanding from the mid regions of Nepal to some other altitude, some lower altitude also. So that is adding also further challenge. And one new challenge that have been added not only in the geography change is also new challenge is also it is now being expanded from pan uveitis to involvement of adnexal tissues. Like this time we had cases where we had SHAPU manifestation along with scleritis. So it was very challenging for us to treat and manage and gain the visual recovery. Even we had cases where the patient of SHAPU they presented with panophthalmitis. Now, what do we label them? Seasonal hyperacute panuveitis with panophthalmitis, or what to say? So the disease is expanding in geography. It is expanding within the eye also. So these are the challenges, and many of the time when we undergo vitrectomy in these patients, we need to do lancectomy for a clear vision. And these the childrens where we are keeping primary intraocular lenses, they are coming with more complications. So we advise avoiding the primary intraocular IOL implantation in a child of SHAPU where which undergo lancectomy along with vitrectomy. So our expansion on the entomological side are we have now doing the GIS and GPS tracking system. So we are trying to identify the moth habitat in that particular region so that we can make the people of that region alert. We are also undergoing the specific CT microscopic test in different forms. We're trying to understand what are the biochemicals, what are the toxins that are present in the moth hair or the CTs, and are they the same that are present in the ocular surface or ocular samples. Moreover, we are trying to be we are trying to undergo more higher level of study by doing the high resolution mass spectrometry analysis of those moth and CT, and also the metabolomic study. And one more thing that we are attempting is to detect the moth outbreak prediction model. So which are the areas that will be prone for moth prediction in the next few years? We are trying to develop those moths so for the prevention purpose. When we extracted the CTs from the moth, then we did this examination under the electron microscope. We found that these have the sharp end, they have a hollow tissue, they have the barbed end. So this sharp end and the barb, they can cause the micro trauma in the intact ocular surface, and then this fluid that are present inside this uh, they can lead to the cascade of inflammation. We even took those CTs to the scanning electron microscopy, and we found that structures are quite similar and there are presence of some fluid inside this CT. So we are undergoing further tests to know what are the fluids that are presented. And we even tried with the moth prediction modelings. Where would be the SAPU danger outbreak danger zone in Nepal in the next 30 years. Uh, so this we found that this disease now would be expanding because of this ongoing global climate change. It will be now expanding towards the western part and towards the tropical part of Nepal also. Maybe it will in this is a prediction done only within Nepal. Maybe in next few years, because of the global change, it could expand outside Nepal, and it could affect other regions of the world also. So we all could be at the threat of getting this disease. We are even running metabolomic analysis, and we have found some of the proteins that are present in those moth cities are related to plant, some are related to humans. So we're analyzing each of these proteins to come to a final result very soon. And one very good thing that we came when we're undergoing this One Health study in SHAPU is that we could find the biocontrollers for this moth. We found that the wall lizard, so as I said earlier, we all live in an ecosystem. So for this moth, they come close to light, white light. When they come close to white light, they feed on the small insect that are present around the white lights. They're coming close to white light is also for their purpose of their reproductive system. But this white moth, when they are coming close to white light, the wall lizard are also close to those white lights. And this white moth, they prey upon this lizard, they prey upon the moth, and this is how the ecosystem is going on, and the we all fall upon this ecosystem. So when I said that we started our journey with with the knowledge of 10 papers, right now we have expanded with the publication of 25 papers, and a few 5-10 papers are under review in various journals. And with this expansion in knowledge, we have presented SHAPU in different platforms. Now it's being a more common name for our global and our international colleague also. And even the Nature, they paid attention to this disease and published in their journal. And one line that I would like to quote is that in this era of global connectivity, we need to conjure a consortium of global mind and resource so that we can decode and defeat SHAPU very soon. And it's been now 50 years journey of SHAPU. Identified in 1975, now we are in 2025. So we had to, this year is very precious to us, and we are trying to make this year very furthermore precious by doing a lot of works in SHAPU in 2025. And our first contribution to it is the publication of 50 years review paper. That is a half a century quest for SHAPU. And thanks to Dr. Rupesh and his team for this collaboration. So now, finally, the government has also taken the ownership for this disease, and now we are working very closely with the government. And government has involved this disease in its national surveillance system like EWARS and SORMAS. So every case of SAPU that gets in who are affected in Nepal is now reported in the national system. And we are going to various part of Nepal, spreading the awareness and preparing the people for the 2025 SHAPU outbreak. We are very hopeful for collaborations with international UBIHS forum like RUSC, IOIS, so that our international colleagues can help us to undergo future research in this area. And I was very lucky enough to be the part of the launching of Proton in 2024 in Singapore, where I had privilege of meeting this esteemed team at Singapore. With even after this, some of these works, we still are surrounded by few mysterious buzzes in SHAPU, like whether it's an infective or immunological cause. If it's really an infection, which is the sole organism which can be blamed, which virus or bacteria or fungus. If it's an immunological cause, which are the signature cytokines, and how the moths are actually causing this SHAPU. So that relation has to be studied. Why mainly children's, why unilateral, why recurrence? We are yet to find the answers for these questions. And why it mainly reported from Nepal, I think I almost answered, like there has to be a combination of all these bioclimatic variables, the local landscape, the ecological dynamics that is quite fit for this moth and this plant in Nepal. That's why it's reported mainly from Nepal. But now the variation in their presentation are adding more mysteries to us. So maybe we need CIDs and Sherlock Holmes to solve these SAPU mysteries in future. Anyway, we are doing our best brainstorming to explore SHAPU mysteries and to work under one health system. So these are the photographs of the SHAPU warriors with whom we are currently working. And the greatest warrior that I believe is Dr. Madan Upadhyay. He was 35 years when he first coined the disease. Now he's 85. This is the photograph taken yesterday. He's still actively working for this disease, and we're working under his leadership. so that our fight is against SHAPU to reduce the SHAPU blindness, prevent the cosmetic disfigurement, and to maintain the quality of these children who have the long years to live ahead for their family and their nation. So I end up with this picture. This is the child of SHAPU who lost his one eye, but every day I used to examine his eye during his um, this during the time he was having disease, and he said that one day I want to be ophthalmologist like you, and I want to examine your eye. And he tried examining my eyes, and that gave happiness to him. But I am very sorry to say that we could not save the sight of his eye, and we couldn't save the cosmetic disfigurement in his eye. So we are fighting so that this does not happen again. So with all the teamwork, the One Health in SHAPU is moving ahead, and hopefully we'll reach our destination very soon. I thank you all for your patience hearing. Thank you, Proton, and thank you to Dr. Rupesh once again.
Thank you so much, Dr. Ranju. Really inspireful and impressive talk. I think this is a great challenge, and you are ongoing on this in a very good way. So congratulations, and thank you so much for sharing this amazing talk.
Thank you.
So thanks, Dr. Anju. I think very inspiring. And one common thing between SHAPU and myself is I'm also going to be 50 years. So I'm also running in 50 years. So I also was born in 1975, like SHAPU, and I'm going to be 50 this year in November. So again, like your peak of the SHAPU will come there, and probably peak time will be November. So I'll be anyway. So I think very very good work and a really good thing. So let's see if from the audience we have anyone will like to switch on his video, unmute themselves, and please ask question to Dr. Anju directly. And Professor Madan Upadhyay is there as well. Before I invite Dr. Madan Upadhyay to please share some of his pearls of wisdom with us, but maybe from the audience, anyone will like to comment anything or ask anything, please feel free to ask. Anyone? Quite a heterogeneous group is there on this call, but that's good to have. So while people are still thinking about a question, maybe if I can invite Professor Madan Upadhyay to please share his pearls of wisdom about his journey and about his inspiring thing to mentor Dr. Ranju and all the other team in this amazing work. Dr. Upadhyay, sir, if you can share some of the things with the audience here, please.
Thank you, Dr. Rupesh. Before I do that, I would like to take this opportunity to congratulate you in advance for being 50. There's a story that goes back to my first attendance of Asia Pacific Academy of Ophthalmology in Indonesia, and Dr. Lalit Agarwal was there. And then somebody had just reached 50 then, and then Professor Lalit Agarwal, a towering figure as he is, he said, you have now matured. So anyway, you matured very long ago. That's just but a story. The point that I would like to make is there is a system of control within the nature itself. For example, you saw slides of photographs of moths being eaten by lizards. And the other way the nature controls it is the female moth, after giving birth, dies. So that's one way for the nature to control. The whole problem is because we have tempered with the nature, tried and kind of literally destroy it, although not everybody is convinced about that. But so we have to find some biological means of controlling it. Unfortunately, I mean, lizards could be one, but unfortunately, the population of lizards themselves is going down. Again, a vagary of climate change. So this is the kind of dilemma that we are in. What do we promote? Biological methods, good. There are lizards are available, but then the population is declining, and which would mean moth population would increase. Now, to localization, there are important reasons which Ranju mentioned, but a disease always begins as in a small country. WHO reports origin of something in one place and it gradually becomes global. Our fear is, and that fear is borne out by changes in the pattern, the areas that are in geographical areas where they are seen. We suspect, and actually I'm afraid, that one day this disease may spread to contiguous regions and far off regions. And I think to ignore it as a one country disease would be a mistake. And this is why Ranju is trying to globalize it, trying to attract more minds, more than money. Of course, money is needed, but I think we need more minds, and minds which are open. See, as ophthalmologists, we always work in a closed loop. We just have to come out of that. And that's what we did. In fact, we did that very beginning, but as a novice, not having had any kind of training in education about animal experiments, we did some animal experiments, which unfortunately, and I suppose inevitably had to fail. They failed. And now to reproduce the disease, something that we need to do is how do we reproduce disease? How can we reproduce disease in laboratory models? Because experimenting on humans is becoming more and more difficult and is not ethical. So development of an animal model, or these days cell cultures are becoming available. Something like this, producing the disease, reproducing the disease, and studying in either animals, models, or cell culture. I think that would be the direction that we should be taking, and we just like to hear from the audience here what else comes to their mind, and how would if they were the ones who involved in it, how would they proceed? So thank you very much, and this is about all that I have to say.
I think you left the audience, sir, with $1 million question. I hope we have the answer to your pursuit. But I'm sure the way you and Dr. Anju have been pursuing this thing, we will be able to kind of come to somewhere there. But maybe made a good progress, but I'm sure there is still a long, long journey to travel. So let's see how it goes. Carlos, any final points before we give a vote of thanks to Dr. Anju?
Just it happens in my mind like one question, and I'm not sure if you had been like try to evaluate like this type of epidemiological studies where you collect the mods in the areas of where the children lives and try to not only makes metagenomics to the children's fluids, but also to the mods and their specific parts. Maybe this correlation could assess also a good profile in terms of good profile, no, but a good association and causality things that could give more inputs on probable etiological microorganism that could be responsible of it. But this should go like in the way of this way of thinking of the biology. But I completely agree, and I'm very impressed and very happy to listen and learn from you.
Thank you, Carlos. Yes, we are undergoing the metagenomics of the moths and its parts, and we are also comparing the same with the ocular samples also. So hopefully we'll have the results very soon.
I will be happy to read that paper. Thank you so much, Doctor.
Thank you, Carlos.
Thank you. Carlos, you can propose vote of thanks.
Sorry, Prof.
Can
My
propose vote of thanks to Dr. Anju. Okay,
I don't
we'd
know.
like to thank Dr. Anju for illustrious lecture and all the phenomenal work she has done in the field. And thanks again for taking out time and educating us about this rare disease. I'm sure the audience would have benefited by understanding or by knowing about this disease. And we do acknowledge that there is still a lot of work to be done. But I think you're leading the way. And once again, as I say, that leading the way from a country with limited resources, particularly related to research, I think that something is really phenomenal. And we look forward to learn more from you, Dr. Anju, and the team. And thanks once again for all the time. And thanks to all the wonderful audience for spending once again your Friday evening. And we look forward to see you on the last Friday of March again for yet another lecture series from Proton. Thank you so much.
Thank you,
Bye.
Proton, and the whole audience. Thank you so much. Bye bye.
Thank you. Bye. Bye.